Welcome back, HSC Bio Crew. I'm Simon, and today we're diving into recombinant DNA and all the transgenic goodness that goes with it.
And I'm Lachlan, recording from the laboratory as usual. If you're cramming this the night before an exam, chuck the highlighters down and just listen for a bit.
All right, foundations first. Recombinant DNA is just DNA that's been stitched together from different sources. So you might have a human gene joined into a piece of bacterial DNA. That new combo is recombinant DNA.
To do that stitching, we need a few tools. First one, restriction enzymes. These are like super picky scissors. They recognize a specific base sequence and cut the DNA exactly there.
And the way they cut often leaves what we call sticky ends. Picture the double helix being cut in a staggered way. So you've got short, single-stranded overhangs with exposed bases. Those overhangs can base pair with matching sticky ends from another piece of DNA cut by the same restriction enzyme.
Then we need ligase, full name DNA ligase. That's the glue. Once the sticky ends have matched up by base pairing, ligase seals the sugar phosphate backbone so it becomes one continuous piece of DNA again.
We also talk a lot about plasmids and vectors. A plasmid is a small circular piece of DNA found in bacteria, separate from the main bacterial chromosome. Because it's small and easy to move between cells, scientists love using it.
And a vector is any vehicle that carries DNA into a cell. A plasmid can be a vector. So can a virus, or that agrobacterium bacterium you'll meet with Bt cotton later. If it delivers the gene, it's a vector.
Cool. Let's walk a classic HSC diagram sequence, making recombinant DNA using a human gene and a bacterial plasmid. This is one of those draw and label the steps questions.
Step 1. Isolate the required gene from human cells. Maybe it's a gene for insulin or a clotting factor, whatever. You extract the DNA and use a restriction enzyme to cut out just that gene.
Step 2. You remove a circular plasmid from a bacterial cell. That plasmid DNA is also cut open using the same restriction enzyme. That's important. Same enzyme, matching sticky ends.
Step 3. The sticky ends of the human gene and the sticky ends of the plasmid meet and anneal. That just means the exposed bases hydrogen bond and match up, ATCG.
Step 4. DNA ligase comes in and joins the sugar phosphate backbones, sealing the nicks. Now you've got recombinant DNA, a plasmid containing a foreign human gene.
Step 5. That recombinant plasmid is put back into a bacterial cell. The term your textbook uses is transformation. The bacteria take up the plasmid and start copying it when they divide.
Step 6. As the bacteria reproduce, you get multiple copies of the desired gene. Later, that gene could be inserted into an egg cell of another species, so it becomes part of that organism's DNA after fertilization. That's where transgenic species come from.
All right, so that's the cut and paste bit. But how do we actually get DNA into cells in the first place? The syllabus gives you 4 main gene delivery methods. This is a classic list and describe short answer.
Number 1. Microinjection. Imagine using a super tiny needle to inject DNA straight into the nucleus of a single cell, usually an egg cell. It's literally what it sounds like, microneedle direct injection.
Number 2. Biolistics, also called the gene gun. I love this one. DNA is coated onto microscopic particles like tiny gold balls, and those are fired into plant tissues at high pressure. It's like a DNA shotgun, but controlled.
Number 3. Electroporation. Here, you zap the cell with a short electrical pulse. That makes the cell membrane more permeable. Little temporary pores open so DNA can slip in, like briefly opening extra doors in the cell wall.
And number 4, using a viral vector. In this case, a virus, or a plasmid or liposome, is engineered to carry the desired gene. The virus infects the cell, and instead of dropping off its own harmful genes, it delivers the therapeutic gene. In gene therapy trials for cystic fibrosis, they've even used aerosol delivery, a nasal spray of viral vectors.
If you're revising, try making a table, method, how it works, and one example use. That sort of structure screams easy marks in HSC short answers.
So now that we can make recombinant DNA and deliver genes, Let's talk transgenic species, because that word pops up everywhere in this module.
A transgenic species is one that's been created by moving a gene across species. So we take a gene from one species and insert it into the DNA of another species.
Key exam point. That gene has to end up in the germline genome if it's going to be inherited. So it needs to be in a germline cell, like an egg, sperm, or early embryo, not just in some random skin cell.
Right. If the gene only goes into body cells, somatic cells, it might help that one organism, but its offspring won't get the gene. When we're talking transgenic cotton or sheep, we're talking changes that pass to the next generation.
Speaking of cotton, let's hit Bt cotton, because that's your set agricultural example. Traditionally, cotton farmers had to spray heaps of broad-spectrum pesticides to kill caterpillars like Helicoverpa zea. Over time, those caterpillars evolved resistance through natural selection. Absolute nightmare for farmers and not great for the environment.
Scientists solved this by borrowing a gene from a soil bacterium called Bacillus thuringiensis. That's where the Bt comes from. This gene codes for a protein that's toxic to certain caterpillars, but harmless to humans and most other animals.
But here's the cool bit. In the plant, the Bt protein is made in an inactive form. When a caterpillar eats the cotton, its gut converts that protein into the active toxin, and the caterpillar dies. So the plant basically defends itself.
Process summary. This is a great diagram to practice. First, normal cotton seedlings are cut into small pieces and grown on solid medium to form calluses, clumps of undifferentiated cells. Then those callus cells are moved to liquid medium with hormones, so they develop into cotton embryos.
Next, scientists cut the Bt gene out of the Bacillus thuringiensis genome using restriction enzymes. That Bt gene is recombined into the genome of another bacterium, Agrobacterium tumefaciens, which naturally injects its DNA into plant cells. That makes it a handy vector.
Then, the cotton embryos are dipped into a solution containing Agrobacterium carrying the Bt gene. The bacteria inject the Bt genes into the cotton cells. Those embryos are grown in tissue culture, then onto solid medium, then into pots and glasshouses. The resulting plants are a transgenic species, cotton with a bacterial Bt gene in their genome.
In the paddock, this means farmers spray far less insecticide. They might still use narrow-spectrum sprays to knock out mites and sucking insects, but beneficial insects like ladybirds and wasps mostly survive. So, less chemical use, lower costs, less selection for resistance, compared with hammering everything with broad-spectrum sprays.
But resistance is still a risk. To slow it down, farmers plant refuge crops, like pea plants, near Bt cotton. Caterpillars feeding on the refuge don't get exposed to the Bt toxin, so moths with any resistance alleles keep mating with non-resistant ones. That reduces the chance of building up lots of double recessive, fully resistant caterpillars.
Another agriculture example from your text is high cysteine alfalfa. It's been genetically modified to produce higher levels of the amino acid cysteine. Sheep that graze on it grow higher quality wool. Researchers are also looking at inserting that high cysteine gene straight into sheep, which would make the sheep themselves transgenic.
So, pros and cons. On the plus side, transgenic crops can reduce pesticide use, improve yield, and help crops resist pests or diseases that used to wipe them out. That can be a lifesaver for farmers and for food security.
On the minus side, there are environmental worries. If everyone grows the same Bt cotton variety, you can lose genetic diversity, heaps of fields with very similar genomes. If a new disease hits that variety, the whole lot can go down like a dodgy batch of prawns at Christmas.
There's also concern about non-target species and ecosystems, like what happens to insects that rely on the old weed species around cotton fields? Or what if Bt genes move into wild relatives? That's why you'll see those debates about biodiversity and long-term impacts.
If an exam question says, evaluate transgenic crops like Bt cotton, you want to hit reduced pesticide use, refuge crops to manage resistance, benefits to farmers, and potential biodiversity loss in the long term. Tick all those boxes and you're laughing.
Let's swing across to medicine now, because the same tech underpins some very cool treatments.
First one in the syllabus is gene therapy. In gene therapy, a healthy copy of a gene is inserted into defective non-germline tissue in a developed plant or animal. Non-germline just means body cells, not eggs or sperm.
Because you're targeting non-germline cells, the change helps that individual, but is not passed on to the next generation. That's a key contrast with transgenic species, where we deliberately hit the germline so it is inherited.
Often, gene therapy uses those viral vectors we talked about, like adenoviruses or liposomes, to carry the healthy gene into lung cells or blood cells. Trials for cystic fibrosis have even used aerosol sprays, so patients just breathe in the vectors.
Then we've got fully transgenic animals used in medical research. Knockout mice are a classic. Researchers inactivate, or knock out, a specific gene by replacing it with an artificial DNA sequence at the blastocyst stage of development.
Those modified stem cells are put into a blastocyst, implanted into a female mouse, and eventually you get offspring that are pure breeding for the knocked out gene. If those mice show, say, higher cancer rates, you can infer that the missing gene was a tumor suppressor.
Because mice share many genes and organ systems with humans, they're super useful models for diseases like cancer, diabetes, obesity, Alzheimer's, and Parkinson's. It lets scientists test potential drugs and study disease progression in a controlled way.
Another medical example you need: transgenic sheep in Australia that have been given a human gene for a blood clotting factor. People with haemophilia lack this factor, so scientists express it in sheep milk, extract and purify it, then use it as a treatment. Cheaper and safer than relying on human blood donations.
Speaking of safer, let's talk recombinant vaccines, especially the hepatitis B vaccine. Instead of using the whole hepatitis B virus, scientists pick the gene that codes for one surface antigen, the bit your immune system needs to recognize.
They isolate that surface antigen gene and insert it into a plasmid. The plasmid DNA is cut with a restriction enzyme. The viral gene is joined in with ligase, so again, recombinant DNA, and then that plasmid is inserted into a yeast cell.
Those modified yeast cells are incubated, so they multiply and produce stacks of the hepatitis B surface antigen. The antigen is then extracted and purified to make the vaccine. No whole virus, just the antigen.
Because you're not dealing with live or whole viruses, recombinant vaccines have a low risk of side effects and are relatively cheap to produce. That makes them great for large-scale programs, especially in developing countries.
Last big medical one, xenotransplantation. That's using organs from other animals for human transplants. Demand for human organs is way higher than supply, so researchers are looking at organs from transgenic pigs.
These pigs are engineered to have human complementary surface markers on their cells, basically proteins on the cell surface that make the organ look more human-like to our immune system. That helps inhibit organ rejection when the organ is transplanted.
So potentially, you could have transgenic pig hearts, kidneys, livers, even corneas available for patients stuck on long waiting lists. Lots of ethical debate, but scientifically, it's the same recombinant DNA toolkit in action.
All right, quick HSC-style recap to finish. 3 big ideas to compare: recombinant DNA, gene therapy, and transgenic species.
Recombinant DNA is the technique of cutting and joining DNA from different sources using restriction enzymes, sticky ends, and ligase, often in a plasmid vector. It's the underlying technology, the toolbox.
Gene therapy uses that toolbox to insert healthy genes into non-germline cells of an individual to treat disease. It induces genetic change in somatic cells only, benefits that person, but doesn't change their offspring.
Transgenic species are organisms whose germline genome has been permanently altered by adding a gene from another species, like Bt cotton, high cysteine alfalfa, clotting factor sheep, or those knockout mice. The change can be inherited, so it affects future generations and overall genetic diversity.
If a question asks about current genetic technologies that induce genetic change, you can name these, explain the process briefly, and then talk purpose: agriculture, medicine, or research.
All right, that's plenty for one study session.
Yeah, hit pause, grab a Tim Tam, maybe sketch one of those plasmid diagrams while it's fresh.
We'll catch you next episode for more HSC Biology Brain Fuel.
Cheers, everyone, and good luck with the study. See ya!